Hi, y'all. This is the small intestine notes. We're going to start with the general functions of the small intestine, where most digestion occurs. Specifically, the mechanical or propulsion of chyme. Chyme, if you recall, is the substance that comes from the stomach that's mixed in with a bunch of enzymes, a bunch of digestive juices, and some of that acid from the stomach. So there's also a chemical component of digestion that I'll speak on later. Lots of enzymes involved in the small intestine, and practically all absorption of the nutrients occurs here into the bloodstream. And a part of that is water absorption as well. So water is mostly absorbed at the small intestine too. So some fast facts. The small intestine is about approximately 7 meters long in a living person, which is quite long. So the name small intestine comes from the width of the structure. And it has a surface area 100 times greater than your skin. So if you think about your skin, it stretches throughout your body. 100 times that surface area would make up the surface area of the small intestine. So there's something peculiar about the small intestine that we'll talk about later that increases its surface area. The 3 parts of the small intestine we'll discuss in a moment. We kind of discussed this in the pig practical section of our unit. So the first is the duodenum. The duodenum comes right off the stomach, the jejunum, and the ileum. So my hint I've given in the past is DJI, playing at your favorite club, Club Flores. So DJI, duodenum, jejunum, ileum is the way that we can remember that. So notice how the small intestine in the human is tucked pretty nicely inside, more medially, and the large intestine kind of frames on the outside. Different from the pig, where the pig, it was just kind of collected on one side, and then the large intestine was collected on the other. So the structure of the small intestine from the proximal or like stomach end to the distal end. So stomach end, first, we start with the duodenum. You might hear an occasional dog creeping by. Bear with me. Most of your chemical digestion happens in the duodenum. It's the first 10 inches of the small intestine. So what is happening here? If you look at our inset picture, you see the pancreas is kind of like right there where the duodenum is, and the bile duct. If you recall, the bile duct leads to the gallbladder in the pig, same for the human. So this intersect, excuse me. This intersect is called the hepatopancreatic ampulla, also known as the ampulla of Vater. It comes from a German scientist or a German name. So that's where like that name comes from. Anyways, this intersect, if you zoom in further, this intersect is where the bile duct and the main pancreatic duct fuse together. It opens into the duodenum at the major duodenal papilla. So you can see it here. It's like this little, almost like it's a sphincter, right? Because it's essentially like where it like pinches off. And this is where the flow of bile and pancreatic juices is like controlled into the duodenum. The sphincter, like I said, you can kind of see on my second picture here, the sphincter. It's that ring of muscle that controls, it's like a doorway. We talked about sphincters in the past, the cardiac sphincter, pyloric sphincter, lower esophageal, all that stuff. Sphincters are rings of muscle that are controlled, and I tell you that it sometimes reminds us of like a doorway. Okay. So that is the duodenum. It has this nice little intersect. And so what is happening? Like, why do we need bile and all that stuff? That's going to help us with our digestion process from these accessory organs of the gallbladder and the pancreas. So the next structure is going to be the jejunum. The jejunum, if you recall, is the middle section. And that's where most of your nutrient absorption is taking place. So chemical digestion first, mainly because all of those juices and all those good enzymatic things are happening first. Then the nutrient absorption, is taking place in the second part of the small intestine. And that part is a little bit longer, right? 10 inches for the first section, the duodenum, and then 3 feet for the second section. The last one is ileum. So that's where our acronym DJI comes from, for your helping organize your thoughts of the small intestine order. The last part of the small intestine is called the ileum, and it absorbs most important vitamins like vitamin A, B12, folate, vitamin E, D, and K. It's the last 6 feet. So it's like the longest part of the small intestine. And this section leads to the large intestine. So there is the part where it joins the cecum. That's that weird, strange bag-looking structure we saw on the pig. Okay. For us, the cecum is the first section of the large intestine area, and that has its own sphincter as well. So I'm not sure if I have a picture of that here, but I will have it in the large intestine notes. So for now, you could just say that like right here in this area is where it's called the ileocecal sphincter. That word comes from ileum, which is the last part of the small intestine, and then cecal is like cecum. So that is how we can know where the sphincter is and like what it's doing. So it's probably controlling the flow of things going from the small intestine to large intestine. Okay, the histology. So this is the part where we kind of look at deep diving into the cells. So there are inside the small intestine, if you were going to look at the layering, it's a lot like the layering we saw earlier. So go back to like just the first set of notes that we did. The layering is pretty much the same. Notice how inside, this is where the food is touching, right? In this case, it's called chyme. And as you make your way out more superficially, you get from the epithelium, muscularis mucosae, submucosa, and then muscularis, so in the outside. Okay. There are circular folds that are essentially like lining the epithelium. So if you take a look at the small intestine cross section, I have it here. This is also in your worksheet that you've done. So you've seen this before. If you take a slice or a cross section of the small intestine, you'll see that there's these little speed bumps. This is making sure that you're not driving your chyme like you're driving your Porsche. Like you don't want it to go sliding through the small intestine really, really fast. You want a really slow process, because slow means you're going to suck up all the nutrients and get all that chemical digestion happening. So the speed bumps are there. We call those the circular folds, to slow down the process so it doesn't go speeding by. Okay, so circular folds act like speed bumps to slow down the chyme. So they are deep ridges in the mucosa and submucosa. They run from the proximal duodenum to the middle of the ileum, and they increase the absorption by slowing the movement of chyme. So that's like one part of the histology here, the circular folds. You probably won't see that in any other part of the digestive tract. So that's something new. Villi. Villi are these little projections in the intestinal wall. So they are finger-like projections in the intestinal wall. So what do they do? Why do we have these little fingers found in the intestinal wall? Well, they allow us to increase our surface area. of the small intestine. So let's imagine that this is a villi. It looks kind of like this. It's a finger-like projection and it's found in the intestinal wall. Each one of those has little projections on them called microvilli. So the microvilli are in orange and the blue thing is the villi. So what I'm trying to tell you is that we are increasing surface area by essentially taking the surface and making it so that there's more surface area to volume. So we're increasing surface area to volume ratio. That's what we're doing here. And we want to do this so that we increase our absorption amount. Okay, so if we look at the picture, if you look closely at the circular folds and zoom in, you'll see the little projections called villi. And if you zoom in on those projections, the projections have projections. So the villi have villi. We call those microvilli. And so this allows, here's the microvilli. You can see them here. So you can see that the surface area increases so drastic. I told you that the surface area in the small intestine is 100 times the skin. So it's pretty remarkable. And that's how you get all your nutrient absorption for the most part. So these villi contain blood vessels and these things called lacteals. Okay, so if I look at this villi that I drew, and this one is the picture, you can see that there's blood vessels here. We call them capillaries, blood capillaries. And inside is this yellow structure called a lacteal. So blood vessels, what they do is they absorb sugars. Oh no, where'd it go? They absorb sugars, so your carbs and amino acids. So 2 macromolecules there, carbs and amino acids are absorbed there. So then what's left? Well, the third macromolecule, major macromolecule, is lipids, fats. So fats are going to be absorbed through the lacteals. So let's say lipids. Okay, so you can see the structure, the lacteal inside, and it's kind of surrounded by these blood capillaries. So all of your major macromolecules are absorbed in that one place in the villi. There also is goblet cells that secrete mucus in these areas so that we can reduce friction. Microvilli, like I said, are bordering. So you can see these little things here, and there's like little arrows, and then it says, hey, let's look closer at this border of the villi, and you can see that the villi have microvilli. So like I said, increasing surface area by like a lot. Okay, so we call that microvilli the brush border. They are extensions of the plasma membrane of the epithelial cells of the mucosa. There are 200 million per square millimeter, so there's quite a lot. So that helps us increase our surface area for absorption of nutrients and water. So if you have celiac disease or somebody you know suffers from celiac disease, I'll go ahead and write that down. Celiac disease is a disease where you have a malabsorption disorder. So the lining of your villi is something's wrong with it. So then people aren't getting the nutrients they need. And so that's something you can, pause and research if you'd like, because I think that's pretty interesting. And I know quite a few people personally who suffer from celiac disease that have special diets in order for them to absorb nutrients properly. So in the intestines, there are more crypts. So we talked about the crypts when we talked about stomach nodes. So these are a little bit different in the fact that they are in the small intestine. They do produce intestinal juices, which is an alkaline solution with bicarbonate ions to help neutralize the acid, right? So like we're trying to bring the pH back up to neutral so that eventually whatever's left when it's exiting the body, it isn't too acidic. So you produce about it depends. So it's about 1 to 2 liters of these intestinal juices a day. And in these crypts, you can see here some of the cells that you would see. And part of that is also some hormones and some other stuff, but we'll start here. So the first thing we're gonna say is the intestinal gastrin. What this is it stimulates parietal cells to release hydrochloric acid and release pepsinogen. Okay, so this gastrin is made by the G cells of the stomach. And so it will stimulate those parietal cells and the release of two things, hydrochloric acid and pepsinogen. So ultimately, that will help us break things down and help us break down proteins. Or yeah, because pepsinogen is the inactive form of pepsin. CCK is next. CCK stands for cholecystokinin. It's a very long word that just means it helps us stimulate digestion of fats and proteins. So what it'll do is it'll help promote secretion of pancreatic juice and bile and the slowing of gastric motility. Lastly, in the intestinal glands, secretin is produced. And so that is going to help us promote the secretion of pancreatic juice and bile, thus helping bring back the pH and promoting bicarbonate ion release. I'm so sorry, guys. My sickness is just not going away. Okay. So, digestion mechanically. So we talked a lot about chemically in terms of like what's happening in the glands, the histology. We talked about the structure of the small intestine, but mechanically, what is happening in the small intestine? So right here, I have the word segmentation. Segmentation is when contents are shoved back and forth. They're segmented into smaller pieces, and that forces the chyme against the wall for absorption. So I want you to pause here for a second, and I want you to Google a picture of segmentation and add it here to your notes. Like, what does this mean? Segmentation in terms of digestion. I want you to put a picture of it in your notes because I know there's some that you can Google. And so that will help you kind of visualize what does segmentation mean? Because that is a mechanical part of digestion. Peristalsis is different. Peristalsis is pushing the food through. It's more of like a forced like push through versus segmentation is more like mixing of food and like segmenting it into pieces. So like I said, I would find a picture on Google, pause, and come back in a second. When motility in the ileum, which is that last part of the small intestine, increases, the ileocecal sphincter or valve will relax. And that food will pass into the cecum, which is the first part of the intestine. So when you have more and more of that movement in the ileum, the door will open to the large intestine. That's what that means. Chemical digestion, the liver will make bile, and thus that's why we talked about the bile duct, and that's why we talked about the valve and all that stuff earlier, the hepatopancreatic sphincter, that section of the notes today. So this bile that's made in the liver is stored in the gallbladder when it's not used. But when it is used, the bile will flow from the gallbladder through the bile duct into that special intersect right up here that I talked about earlier. And so what does the bile do? Like, why do we have all of this controlled things happening with bile? Well, bile helps us break down fat easier for digestion because fats are kind of hard to break down. I mean, you've seen a greasy pan, right? It requires soap and a little bit of scrubbing in order for us to break down fat. Well, just like dish soap, we have bile that emulsifies or actually breaks down the chemicals in the fat. So as you see in the picture, the fat will be emulsified. Essentially, it's going to be surrounded by these bile salts that are found in bile, and those will actually help flip the charges of the fat from non-polar to polar. And so that will allow it to help break down in water. And that's how this works. It's kind of cool, actually, to think about it that way. Other chemical digestion happening is pancreatic juice. So we spoke about pancreatic juice being flowing into that special intersect, like I just said. So pancreatic juice, what does it have? It has lots of water and salts. So that helps with breakdown. It has sodium bicarbonate that helps neutralize your stomach acid, and it inactivates pepsin. So we can no longer, break down protein at this time based on pancreatic juice. Pancreatic lipase. So pancreatic lipase as lipase lipids, it breaks down fats. Pancreatic amylase. Amylase, if you recall, there's another kind of amylase in your mouth called salivary amylase. So it does the same function, it's just made in a different place. Salivary amylase is made in the mouth. Pancreatic amylase is made in the pancreas. Both break down starch. Nucleases break down DNA or RNA. So those are going to be your nucleic acids, a different macromolecule I haven't mentioned yet. RNA. Trypsinogen, procarboxypeptidase, and chymotrypsinogen are all three inactive proteases. So if they're inactive, that means that they're made in the pancreas, but they're not active yet. So those are going to be probably active later when they're needed. So we'll go ahead and say zymogens for now, because zymogens are enzymes that are made somewhere but inactive and then active later. Okay, so they're probably going to be involved in the next couple minutes here. Pancreatic proteases are zymogens. Oh, we already said it. Perfect. They're inactive when they're produced, or they'll digest the pancreas, which means that's when people have pancreatitis. Essentially, that's an inflammation of the pancreas because your proteases are active too early. So in order for them to work appropriately, they have to be zymogens, because then they would eat. Remember how I spoke about how you would just eat yourself from the inside if you don't have very carefully programmed enzymes. So these three are one of that concept. So proteases break down proteins. So that's why we don't want to use them yet, right? We want to break them down later, because we want to break down proteins, right? Because we want to absorb the components of proteins, which are amino acids. So activation of these proteases, these three, is very orchestrated, like very calculated. It happens in two stages. First, we have another enzyme called enterokinase. So we can say it enters, or it's starting the process. It activates this enzyme called trypsinogen to trypsin. So remember, when it ends in ogen, it's inactive. That was our hint. Zymogen, trypsinogen, etc., into trypsin. So this activation of enterokinase activating trypsinogen into trypsin will then cause another chain reaction. So trypsin from the previous step will convert procarboxypeptidase into carboxypeptidase, and chymotrypsinogen into chymotrypsin. And so this whole step thing, this very calculated and programmed initiation of all this is happening in the small intestine epithelial cells. So basically the microvilli we spoke on earlier. So these little things here are the microvilli. So what is happening? Well, enterokinase is like a little, it's just resting on the microvilli. It's there. It's not moving. Enterokinase is not moving. So what happens? Well, trypsinogen will get kind of close. And so when trypsinogen gets close enough, it'll activate, it'll like brush by the enterokinase, the little red dot. When it does that, it activates trypsinogen, which is right here, into trypsin. See? Then that same molecule, trypsin, will be over here next, and that will help activate chymotrypsinogen to chymotrypsin. And we have another activation here of procarboxypeptidase into carboxypeptidase. And so what is this? Why are we doing all these programmed activations where you have to be in the small intestine or else it won't work? Well, we don't want to activate them anywhere else. I can't make that smaller. So you don't want to activate them anywhere else. So that's why they need to stay. All right. So what do those three things do for us? So what does trypsin, chymotrypsin, and carboxypeptidase do? They help digest proteins. That's what they do. Okay, but they're not active until this whole signaling event happens first. So first, chymotrypsinogen is activated by enterokinase, and that'll transform it into trypsin. Trypsin then goes and does the next step. Trypsin says, all right, I'm going to activate chymotrypsinogen into chymotrypsin. And so those three enzymes that are now active will be active and digest proteins. But you don't want protein digesters anywhere else in the body, or else you start eating yourself from the inside, right? So we don't want that. So it has to be very controlled. So here's another picture of what's happening, right? And this has to do with your like thinking of food and it being your stomach. It'll activate some of those nerves to release the pancreatic juice, and the chyme entering the duodenum will activate some other nerve reflexes and nerve cells. And so that will help with some of the hormones like secretin and cholecystokinin, which I mentioned earlier up here. These three. Okay. So this picture is just showing you that like everything's kind of related and it all works together so that you can have a properly functioning digestive system. Lastly, before I let you go, here is a table of all of the enzymes, where they're made and where they work. So where they're made is here, where they work or what they do is here. Okay. So this will help you kind of get your bearing straight of like what we've learned so far. Some of these enzymes in this region, we call them brush border enzymes because they're pretty much hanging out on the microvilli. Okay, for the most part. And then I think we're missing one. I think we can leave secretin out of this, but we'll just stick with this list so far, and I'll help you make a list of all the enzymes you need to know for the exam. Thank you for watching. Bye.